Okay, so I'm going to be talking about branch chain amino acids and leucine. That one is specific, and the signaling response has to do with protein synthesis. Leucine is one of the three essential branch chain amino acids. Like I said earlier, it stimulates the muscle's signaling pathways. It is hydrophobic and is a constituent of most proteins. The purpose of leucine: exercise increases muscle protein synthesis by stimulation of signaling pathways inside the contracting muscle cells. When no source of protein or amino acids is provided and there's no increased availability of amino acids, positive protein balance will not occur. There are two main reasons to intake leucine and other amino acids, and that is to stimulate the signaling pathways and to provide building blocks for new proteins. And one thing that leucine does with the rest of the two other branch chain amino acids is that it activates mTOR, which is, they call this the gain switch, and this basically helps the muscle to receive protein and to grow, basically to get bigger and stronger. Um, Several studies have demonstrated that signaling is increased post-exercise with the ingestion of the three branch chain amino acids. No studies in humans have measured protein synthesis in combination with these measurements. Branch chain amino acids alone can stimulate the signaling cascades. This will not result in increased protein synthesis if the amino acid building blocks are not supplied at the same time. Protein synthesis does not only depend on the activation of protein synthetic machinery, it also is also dependent on its substrate. One of these studies found in 2009, uh, they found that they wanted to demonstrate that branch chain amino acids, their importance for increasing muscle protein synthesis and ultimately the training. Uh, they did a study on rats, and they had the rats run for 2 hours. They observed that the muscle protein synthesis was decreased following the exercise. When leucine was given to the rats, the signaling pathways were stimulated and protein synthesis was increased. But it was only increased to pre-exercise levels, and this situation is very different to what is typically observed in humans. So leucine has a greater effect on rats than it does in humans from this study. That's what they got from it. Um, Researchers demonstrate that the addition of protein to carbohydrates following resistance increases muscle protein synthesis. However, when they added extra leucine to the carbohydrate protein mixture, this did not result in further improvement in protein balance. Branch chain amino acids, in particular leucine, have a role in signaling that enhances protein synthesis. It enhances it. It is unlikely that leucine alone will stimulate protein synthesis post exercise because of the other amino acids have to be supplied as well to serve as building blocks. So there have been people who were studying or researching whether to just take leucine, but according to this study, other amino acids are needed as well to help build muscle. Okay, in humans, leucine in humans, exercise does not decrease muscle protein synthesis like it did in rats in this study in 2009. The leucine did not increase protein synthesis in these rats above pre-exercise level. It merely returned muscle protein synthesis to the levels observed before exercise. Whether leucine would be effective in humans, where such a catabolic state could not be observed post-exercise, it still remains unclear. Now, in the latest research, Luard scientists conducted an experiment using forearm balance method. This method measured the response to IV infusion of a mixture of branch chain amino acids for 3 hours in 10 post-absorptive subjects. 3-hour infusions of branch chain amino acids increased plasma concentrations of all branched chain amino acids. During this 3-hour infusion, muscle protein synthesis was decreased. Balance between muscle protein synthesis and breakdown remained negative. This showed that an anabolic state was not produced and a catabolic state remained. Decrease in muscle protein synthesis simultaneously breaking down during branched chain amino acid infusion can be referred to as decreased muscle protein turnover. Similar results were gathered when the infusion of branched chain amino acids was extended to 16 hours in 8 volunteers, used to determine if chronic elevation of branched chain amino acids stimulates muscle protein synthesis. The results they gathered from this, muscle protein synthesis was reduced in subjects. Muscle protein breakdown was reduced and catabolic state remained. So conclusion from these latest research studies, it can be concluded that branched chain amino acids infusion fails to increase rate of muscle protein synthesis in humans, actually reduces the rate of muscle protein synthesis and muscle protein turnover. Reduction in rate of muscle protein turnover has immense effects on muscle strength. Muscle protein turnover renews muscle fibers and results in increased efficiency of contraction at the single fiber level, even if muscle mass is maintained. Uh, Thank you for listening, and enjoy the rest of the videos. Go Gyrenes.